Okay, so welcome everyone to one of the series of seminars we're giving on the translational impact of our work here at the LSTM. As you'll be aware, one of the three key aims of LSTM is to maximize the translational impact of our work, and translation in this case in a public health sense. And so we have a number of groups, 5 presenters from across the vector department, who are presenting a sort of an evolving narrative, if you will, around our work in insecticide resistance and vector behavior, and how that's been led into real-world impacts on malaria transmission, principally in sub-Saharan Africa, but how it's led to the development of new vector control tools, and how from a mechanistic understanding of insecticide resistance and behavior, we've transitioned through product development into public health impact and policy change on a global scale. So that's what we're hoping to capture today. This is a work package from a number of groups across the school, and what I really want to emphasize, it's very much in association with and in many cases led by partners in the global south. And it's been a research area which has been going on for approximately 20 years within the school. So that's actually why I reflected on, we have sort of quite senior presenters here who are at the outset of these projects, early career researchers. And I also just wanted to emphasize that a lot of this work has been driven by successes of cadres of highly talented and very gifted early career research, and that will be reflected in the acknowledgement slides, and that's why we have this presenter composition for. So we're going to kick off with Dr. David Wheatman in the department, who's going to talk us through some of the mechanistic understanding, and then we'll have, fingers crossed, a sort of a scientific relay where we'll pass on to other members of the department who will talk about their specific areas of interest and how they've moved the story forward. So welcome, and over to you, David.
Okay, thanks very much, Martin. And that's a good start. The slides aren't advancing. Right, sorry. Slides have gone to sleep there. Okay, so it's, I'm going to be starting out the first of the sub-presentations, if I like, by looking at the role that LSTM and of course many others have played in advocacy, specifically focusing on developing the evidence base to show the importance of insecticide-treated nets, but also the threat to that importance that is posed by pyrethroid resistance. So, although use of bed nets for protection against flying insects and the diseases they may carry has been around for hundreds or probably thousands of years, a big revolution really came or became possible from the sort of early 70s onwards with the development of synthetic pyrethroids. So synthetic pyrethroids are have neurotoxins that are fast acting, and really importantly, in addition to, although they're very good at killing insects, they're very, very safe for humans. That means that they can be used to treat materials that are going to be in close contact with people. And this property meant that they were highly suitable for treating bed nets. And this led to the development of bed nets or treatments for bed nets, which were then tested in a number of randomized controlled trials. And the results overall from these were, to the point in the early 2000s, was summarized by the Cochrane Review by Christian Lengler. And the take-home message from this was that insecticide-treated nets can reduce deaths in children by a fifth and episodes by a half. There are a number of trials, but one that I particularly like to highlight here, which was led by CDC and KEMRI and research from University of Amsterdam, was conducted in Asembo Bay in Kenya. And the reason why I highlight this one in particular, I mean, the results from several trials were very consistent in showing that there was a strong impact, was because this probably provided the strongest evidence for a community benefit. And the community benefit was demonstrated because the protective efficacy that was shown by insecticide-treated nets that were present within control compounds was also found outside of those compounds, at least within a relatively short space. And this strongly suggested that people were getting protection even if they were not sleeping under a treated net. And therefore there was a strong community benefit, as in mosquitoes were most likely being killed, and that this reduction in the population as a whole was protecting the wider area. It does indeed suggest a major benefit of the insecticide treated nets, and subsequently these became long-lasting insecticidal nets which were treated for the lifespan of the net, comes from the pyrethroid treatment itself and the killing effect. So the evidence was really crystallised for how strongly the impact of treated nets has been on malaria control in a landmark paper from Samira Bhatt and colleagues, which many of you will probably be familiar with. And this compared the Plasmodium falciparum positivity rates in children at a point of 2000 and 2015. And the heat map comparing the A and the B there, left and right, shows a dramatic decline from 2000 to 2015. But of course, many factors were changing, not just the mass distribution of bed nets. What their geospatial model was able to show was what percentage of that decrease was attributable to bed nets. And it was very major indeed, almost around about 70% of that decrease was attributable to bed nets. So it really demonstrated with very good clarity just how important treated bed nets have been for the decline seen in malaria control. Unfortunately, nothing comes for free, and the fact that it was only pyrethroids that have the required safety profile for treatment of bed nets, or certainly has been for a long time, means that they've effectively been used as a monotherapy. Throughout the time that the mass distribution has been going on, there's been a lot of bioassay testing going on across Africa, particularly using the WHO tube bioassay methodology. And this produced lots of data points, which enabled research from LSTM to put together a time series and demonstrate that whether we look at different types of vectors, so we've got Anopheles gamgai at the top on the left and Anopheles funestus bottom left, there's really quite strong declines in mortality caused by pyrethroid insecticides over time as we move through from the 90s to 2015. And to put this into a spatial and temporal context, Penny Hancock is a member of Catherine Moy's team who's been working with Mike Coleman, Janet Hemingway, and various other people from teams at LSTM, have shown that over time, pyrethroid resistance has both spread and intensified as moving through 2015 through to 2017, with particularly strong resistance in West Africa. And obviously this change over time does certainly correlate with the mass distribution of bed nets, and the up-to-date data for this are presented in the plot on the right. But what the model also showed was that by looking at over 100 potential explanatory variables, and this was particularly strong in the West African component of the model, ITN coverage was really strongly correlated with the changes in pyrethroid, or the variation in pyrethroid insecticide mortality. And this was true both spatially and temporally, and so although based on correlation, it strongly implicates that pyrethroid resistance is being driven, at least in a large part, by the increased coverage of bed nets treated with pyrethroids. So this is based on data from WHO bioassays, and whether this actually reflects an operational change, does it mean that there's a derailment of malaria control, has been a long-running argument that really has required data to address this. So there were a number of studies coming from groups at London School who are working in Benin, who, when they compared bed nets that were treated or untreated, these were bed nets that were quite well worn, so they had holes, they were finding that there was very little difference between the treated and the untreated if they were looking at mosquitoes from a resistant area. And this was in contrast to susceptible mosquitoes. Similarly, work from the AVECNet project led by LSTM was showing just how dramatic the difference in mortality for mosquitoes exposed to various types of pyrethroid treated nets were between the local resistant populations in Burkina Faso compared to susceptible strains, as Kasumu strain. So really, it does indeed appear that the nets are not quite doing everything they should do, at least under these test conditions. But that's not quite the same as finding a major impact within a control situation. And to look into this, the WHO commissioned a major research study, multi-centre study spread across 5 countries, which was aiming to look at what the impact of pyrethroid resistance on the effectiveness of insecticidal tools, particularly long-lasting insecticidal nets treated with pyrethroids, were. And the study involved overall looking at about 80,000 mosquitoes across almost 280 locations across multiple time points. Involved testing about 50,000 children for malaria parasites, and about 40,000 were assessed for clinical incidence of malaria. So really huge study, and again, this was led by LSTM. So the results from this were in some ways quite surprising, I guess, at the time, in that of the 5 countries, within each one there were variations, there were clusters with relatively higher and lower resistance, and the comparison was made between whether there was an impact of this difference in cluster resistance on whether net users were favoured or non-users were more favoured. And the consistent message coming from the study was that even in areas of relatively higher insecticide resistance, net users still had a substantial benefit. So it's suggesting that the personal protective effect that's provided by the barrier net, the barrier from the net, even if the pyrethroid is not killing effectively, is still really, really important. So this was slightly at odds with the very early results that suggest a major community impact from the pyrethroid on bed nets, or apparently so. To look into this further, Tom Churcher led a modelling study in collaboration with Hilary Ranson's group, and this was looking at to make a modelling meta-analysis from experimental hut studies, and these were conducted across a wide variety of different resistance settings and involving sleepers under bed nets. These looked at blood feeding inhibition, which is a commonly recorded measure in hut trials, and also the mortality. Difference being that blood feeding inhibition suggests a measure of personal protection, the bed stopping the mosquito getting a blood meal, whereas mortality indicates community protection, something that's going to suppress the population. And the plot on the right shows how the probability of different events, so probability of a mosquito being killed or blood fed as you move towards the bottom right, or whether they're deterred from feeding and exit the hut in the middle, against the survival and resistance testing. And the modelling results showed really quite clearly that high resistance was required to reduce the personal protective effect, but the community effect can be reduced at far lower resistance levels. And this modelling result really helped to tie up some of the other results that we were seeing, both from the WHO impacts trial and the early results from Asembo Bay. So I'll pass over now to Gareth, who'll be talking about mechanisms of insecticide resistance.
Thanks, Dave. In this second part of the talk, we'll be discussing how and why we need to delineate the key mechanisms of resistance in Anopheles mosquitoes. Could I have the next slide, please? And so the research behind looking into the mechanisms of resistance has been going on for many years within the department and has involved many, many different partners throughout the globe. And many past and present members of the Vector Group have been involved in this work. And all these folk together have been at the forefront of the discovery of these mechanisms in Anopheles mosquitoes. Why do we need to know what the mechanism of resistance is in the mosquito? Well, once you know what the mechanism of resistance is, you can find other chemicals or other approaches to attack these resistant mosquitoes. And so much of the work that's been gone on in department in the past 15 years has been looking at the different mechanisms of resistance. And Anopheles, like most insects, have got four main mechanisms that are involved in resistance. This includes the modification or the evolution of changes in the cuticle, which slows penetration of insecticides into the mosquito. You also get changes in target site, so you get mutations in the target site for the insecticides, which stops the insecticide binding and functioning, but that mutation keeps the function of that protein going, so the mosquito survives. Other work that's going on in the department, so we're looking at all these different mechanisms. More recently, another mechanism is sequestration, and these are proteins produced by the mosquito that are able to bind insecticide before it can reach its target site. So there's some work now going on sequestration. But it has to be said, most of the work to date has involved looking at detoxification, and these are enzymes which break down insecticides. So the mosquitoes can produce enzymes in different parts of the body which can break down insecticides. And this, even though we're different members of the department are looking at all different aspects of resistance and the mechanisms of resistance, we'll focus on detoxification enzymes today. And can we have the next slide, please? And it has been known for a long time that there are three classes of enzymes, three superfamilies of enzymes that are mainly involved in resistance to insecticides. These are glutathione transferases, carboxylesterases, and P450s, which will be a main focus for us today because of how we've discovered their central role in pyrethroid resistance. And so this map is indicating through work with partners how mosquitoes have been examined, their biochemical aspects of the mosquitoes have been examined to look at these enzyme activities. And in many cases where we're picking up resistance, you see elevated activities of these enzymes. And as I mentioned, the P450 enzymes are elevated in many different populations. Can I have the next slide? The problem with studying these super families of detoxifying enzymes is that in mosquitoes, in Anopheles mosquitoes, there are over 100 different P450s, and altogether there's over about 400 different enzymes which may be involved, and they all have similar activities. So if you're trying to find which ones are involved with resistance to pyrethroids in this case, it's quite a hard job to delineate which P450s are involved in that breakdown of pyrethroids. And this slide just is showing some of the different tools that have been used to examine which genes, to try and find out which of these genes are involved in resistance. And like most of science, the paradigm shifts in knowledge really come through technological advances, and the Vector Group and their partners have been quite at the forefront of early adoption of these technologies. So this includes the transcriptomic analysis through initially small microarrays and then whole transcriptome arrays, where you're trying to find out which specific genes are upregulated in resistant mosquitoes, which ones are produced at higher levels. There's also movement into SNP arrays, so looking at single nucleotide polymorphisms. So rather than looking at which ones are upregulated, you're looking for mutations in the metabolic genes that may be associated with the resistance. So you can study the evolution of resistance and see if there's common SNPs amongst these resistant genes. And this lends itself to diagnostics. So once you can identify polymorphisms. You can get diagnostics and go and have track and trace to follow the early production of resistance in the field. We've also developed transgenic analysis and in vitro analysis to look at the function of these individual genes. And finally, with the advent of fairly cheap whole genome sequencing, it's now possible to compare resistant mosquitoes from many different populations to look at individual genes and how mutations or duplications of those genes are associated with the resistance phenotype developing. So all of these tools have been used over a 15-year period to delineate the enzymes involved. Okay, the next slide, please. And this slide shows how the data from multiple different transcriptomic analysis has been used to narrow down the number of, in this case, P450 genes that are involved in resistance. So this, together with much of the genomic work, has enabled us to narrow down the field of potential genes that P450 genes that are involved in pyrethroid resistance. And here you can see numbers of different populations of Anopheles taken from areas throughout sub-Saharan Africa, and then doing transcriptomics to see which ones are upregulated. And what you can find is there's a small subset of P450s that are commonly found upregulated in the Anopheles mosquitoes. This is not to say that these are the only genes that are involved, but these are the key ones that keep on cropping up in mosquitoes from different areas. Could I have the next slide, please? And so from all this work, we can narrow down the potential candidate genes that are involved. And in the next couple of slides, I'll show how we can take these candidate genes and show that they are in fact involved in the resistance phenotype and producing the resistance phenotype. And so this first slide shows in vitro assays to examine the activities of these P450 enzymes. And so this is a quite high throughput technique. And so you can take a whole series of the P450 genes, clone them into E. coli and express the recombinant protein in E. coli, and then mix this recombinant protein with the insecticide, in this case the pyrethroids, and then do mass spec and HPLC to determine do they produce metabolites? Is there a metabolism going on of these pyrethroids? And to summarize many years' work, it appeared that some of the key P450 candidates were CYP6P3 and CYP6M2. These showed high activity against different types of pyrethroids and have become key metabolic resistance candidates. Could I have the next slide, please? To take this further, and so we've shown that some P450s are able to metabolize pyrethroids. We were then able to put these individual genes into mosquitoes, into susceptible mosquitoes, and overexpress these two P450s. So we produce mosquitoes that are originally susceptible but just overexpress one of these two P450s. And so these mosquitoes are producing quite a lot of P450 of, in this case, M2. The CYP6M2, and when we do bioassays on these mosquitoes, we can see that just overexpressing one of these gives rise to resistance in these mosquitoes to permethrin and deltamethrin. So M2 and P3, CYP6M2 and P3 give resistance to permethrin. P3 shows a lot more activity against deltamethrin, whereas neither seem to work against DDT. Interestingly, the P3 also metabolizes a totally different class of insecticides, the carbamates, or at least bendiacarbon in this case. And so we've been able to go from hundreds of P450s in the mosquito to show that a small subset of these are involved in pyrethrosis. Okay. And so what is the impact of this knowledge? So, as I said, one key aspect is the ability to produce molecular diagnostics. And this allows track and trace and exterminate of resistant mosquitoes in an ideal situation. The in vivo and in vitro screening of different P450s allows a pipeline to be developed to screen new insecticides to see if these insecticides are going to be broken down by the enzymes that we know are in mosquitoes already. So we can screen to find the most potent insecticides that are going to avoid these resistant mechanisms. And importantly, because we know the P450s are mainly involved in resistance, if we can block P450 activity, we can overcome resistance to the pyrethroids and extend the use of the pyrethroids. And so with that, I'll pass on to Philip.
Thanks, Gareth. Next slide, please. Having demonstrated the importance of the P450s, this was exploited because the piperon butoxide was a well-known and well-researched potent inhibitor of the cytochrome P450 enzyme. So this meant there was an immediate method to respond with a new type of bed net. Next slide. And pretty quickly, Sumitomo and Vestigard began to produce nets treated with PBO plus pyrethroids. So we're still dependent on pyrethroids, but as Gareth said, we're extending the lifespan through the use of this inhibitor. And interestingly, at the same time this was happening, Vestigard's bednet, the Permanet 3, only has PBO on the roof, which was a decision made based upon work from our department, which showed that in a very crude early experiment, the importance of the roof as a primary location where where Anopheles mosquitoes landed. Next slide. But those crude investigations really didn't tell us enough. I mean, it was important enough to make a decision on using treating the roof only, but we expected there was a lot more information to be gleaned if we could only see more of what was happening around the bed net. So we worked with the University of Warwick engineers to develop this tracking system, which was portable enough that we could use it in a hut and look at the behavior of wild mosquitoes at a bed net in sub-Saharan Africa. And this early work was done in Tanzania. So top left shows the tracking system, and the top right, image B, shows probably about 20-second bursts of activity of a number of mosquitoes actually getting through a hole in the net to reach somebody inside. And the bottom image shows what happens when you track 25 mosquitoes over a period of about 1 hour. Next slide. So how does an ITN work? Well, this movie shows what was really quite revelatory at the time. We had never really realized that the mosquito is in flight the whole time. She's not actually resting on the net. So she's visiting the part of the body above the torso and head, not quite exclusively, but predominantly. And from this, we were able to measure a lot of detail. Next slide, please. So first point here is that a bed net, an ITN, is a human baited trap. Without the human sleeper inside, the net really has no function. It doesn't attract and it doesn't kill mosquitoes. When it is fully functional, the majority of contacts with the bed net are made above the torso or the head of the sleeper. And in fact, this, as I said, as we saw, they don't rest for long periods, but this constant flight activity of relentlessly trying to reach the host means they accumulate contact with the bed net surface. And a fatal or lethal dose is received on a fresh net in about 90 seconds, possibly less in some cases. The mosquito comes in, attacks straight away, and in our test of 1 to 2 hours in duration, the majority of mosquitoes are killed within 10 minutes of arrival at the bed net. Now, the important thing here as well is that the profile of arrival, the pattern of the predominant arrival at the top of the net, you can see by these figures on the right. The treated net on the bottom and the untreated net with the host in the middle. The darker patterns are on the roof of the net in all cases. So while the numbers of mosquitoes attacking are reduced, or numbers of contacts, the attack rates reduce, and the behavior shifts from high contact to low contact modes. The top of the net remains the predominant area of activity. Next slide. And that's shown quite clearly here. So these three images show an untreated, unbaited net on the left, untreated human bait in the middle. We see the huge increase in activity and the dominant activity on the roof of the net, in this case, over the entire surface of the net. And when you put insecticide on the net, the color change shows you, as you can see from the key on the bottom, that the mosquito activity ends far earlier in the 1 hour sequence. So the profile is the same across all three different treatments, if you like. And important also is if you look above the net itself, in the space above the net, there's also considerable activity. And much of what was happening up here were mosquitoes flying back and forth. What purpose, we don't know, but not making any contact with the bed net itself. So we wondered if we could exploit that behavior by simply putting a barrier up there, which they would bump into. Next slide. Innovation seems a very strong word for sticking a barrier in in the path of a moving animal, but that's all it amounted to. So here the top image shows a sleeper with the mosquitoes around the net, and adding the vertical panel, and you can see that on the bottom left, it shows it in situ in Burkina Faso. This was sufficient to really increase the amount of contact the mosquito made with the insecticide. And you can see a couple of tracks there in the middle showing individual mosquitoes, and the left-hand side avoiding it, and then the right, there's a right one where it collides with the barrier and with the net itself. The advantage of the roof of the net is that up here, the sleeper will never contact it. Even in daytime usage, it's out of the way of children. It can be sort of ignored, not entirely, of course, but it does mean you can have a safer net. You could potentially use insecticides that we wouldn't have been able to use previously on a bed net, and potentially new insecticides that might cost a lot more than the existing pyrethroids could be used by treating only the barrier, perhaps. So we did some assays in Liverpool before we moved to a hot trial in Burkina Faso. Next slide. So shown here, we have three treatments. So the green shows a permanent 2, a standard pyrethroid treated bed net with a barrier made out of the same material. The blue one is a barrier made out of neonicotinoid, which didn't work very well, so we didn't continue with it. And the yellow and the brown columns show the yellow is a permanent 2 with an organophosphate barrier. So here we're using a barrier that would kill the highly resistant vector population we were dealing with. And the brown one is an untreated bed net with organophosphate on the barrier only. And you can see straight away that the organophosphate, first of all, is very effective because that's the insecticide to which the population of mosquitoes are susceptible. But what's remarkable in some ways is that the untreated net with the organophosphate barrier is so extremely efficient on the left-hand side in terms of killing effect. And not that we're advocating untreated nets, but it does demonstrate that with the correct or potent insecticide, the barrier alone can do the job of that the entire bed net was doing in terms of killing. On the right side, the right figure shows personal protection. So these were bed nets with holes, and you can see that the permanent 2 with the organophosphate barrier is by far the best, actually improves on the pyrethroid only P2 in green. But you can see that the untreated net with holes in it, that you actually have a loss in personal protection, though this wasn't quite significant. Next slide, please. So working with our colleagues in Imperial, they modelled what the effect would be like if we had replaced the current permanent 2 bed nets used widely in Burkina Faso with the permanent 2 and these organophosphate barriers. And you can see if continuing with the standard permanent 2 in grey, you reach that point in 3 years' time, and the 2 other nets are not really significantly different, but what's remarkable again here is the untreated net actually is slightly but not significantly better than the permanent 2. So again, the untreated net plus effective barrier has the potential to be as or maybe even more effective than the current basic permanent 2, which is in use widely across Africa. So since then, next slide. We've gone to a prototype working with Vestigard, the bednet manufacturer, to produce a permanent 3 barrier net. So this is basically the regular permanent 3 with an additional barrier, a longitudinal barrier running from head to toe this time. And this just contains the same material as the blue roof of the bednet. So the reason we did this rather than use any other insecticides was the idea was that we would get this approved, hopefully, more rapidly by using existing chemistries and not having to worry about registration of new chemistries, but get a concept tested in Africa as early as possible. And with that, I'll leave you to the next speaker.
Martin, put your mic on.
Thank you, Gareth. So hopefully by now you've got an understanding of how the LSTM team and partners have been developing an understanding of how behaviourally mosquitoes intersect with the net surface, leading to this treatment of insecticide and PBO on the top, and also that mechanistic understanding of resistance. But a key component running alongside this was related to advocacy. What we were very concerned about is that we needed to sensitize a variety of stakeholders, sort of WHO and the other normative bodies, industry partners, the scientific and public health communities as a large, to the inherent dangers of insecticide resistance. As Dave spoke about earlier, pyrethroid nets at the point were used as a monotherapy. And so, as we know with AMR, the emergence of resistance is a major threat. And we needed to get the community to start to think about what's the strategy for transitioning. And so we use that with a variety of sort of opinion pieces and also sort of use case scenarios to try and sensitize the community about how resistance could potentially impact public health impacts of malaria. And running alongside that, we were generating the evidence base. So these are a Cochrane review led through the LSTM by Kath Gleave and Hilary and colleagues, and in association with the Cochrane Infectious Disease Research Group. So they were collating the evidence for what actually is the impact of PBO and pyrethroids used in concert on entomological and epidemiological indicators of malaria. And I appreciate the slide on the fonts on this are very small, but the take home here is that they identified around 180, 190 different studies which investigated the use of PBO and pyrethroids on a variety of outcomes, both entomological and epidemiological. And only 15 of those met the criteria for inclusion in this review, and only one of those was actually an epidemiological trial. So the outcome of this review is actually they found strong evidence that in areas of high insecticide resistance, PBO nets resulted in an increase in mosquito mortality and reduced blood feeding rates, and we could infer that it's likely to therefore have an impact on malarial transmission rates. And the one clinical study, which was that led by Natasha Protopopov, which was conducted in Maleba in Tanzania, showed that there was a reduction in malaria prevalence in those communities where PBO nets were used and the control group there was standard nets. So there was some emergent epidemiological evidence of impact. Can I go back one, please, Dave? And so alongside this, you saw earlier the work that Tom Churcher and Hillary's and colleagues conducted looking about use cases for PBO nets. And so the LSTM groups started to work with WHO to develop the evidence and interim recommendations for where we may want to think about deploying these sort of second generation nets. And so the ENTO evidence, the Cochrane review and Natasha Protopoff's trial and the advocacy from some LSTM staff members resulted in a WHO coming out with an interim recommendation that these pyrethroid PBO nets were a new class of insecticides. And that's particularly important because before this point, they were bundled in with nets as a whole. And so there was no incentive for manufacturers to move into that market because there's no sort of differential purchasing power for the two net types. This was only an interim recommendation because WHO stipulated that they required evidence from 2 epidemiological trials of the impacts of PBO before they could move to full recommendation and a sort of full classification as PBO as a new net type which had properties which would delay the emergence of resistance. And so that data was generated from a second trial, and this was conducted in association with IDRC, London School, Ministry of Health in Uganda, UCSF, and it's funded by, you can see the funders across IVCC and the Gates Foundation. And this was a cluster randomized control trial, which was embedded within a universal coverage campaign conducted by the Ministry of Health of Uganda. And so essentially what it's again trying to do is evaluate both the epidemiological and entomological impact of PBO nets versus with standard nets. And the primary outcome here was looking at parasite prevalence in children between the ages of 2 and 10. We also looked at anemia rates in a subset of children, and also vector density as a sort of a corollary of hopefully the epi impact we were hoping to see. So it's notionally a 4-arm trial, though it was powered for 2 arms, so essentially a PBO versus non-PBO comparison. And I think it's important to point that the randomization process, we stratified it both on the east and the west of the country because work led through by Dave Wheatman had identified P450 mediated resistance in the east of Uganda, but we didn't really know much about what was going on in West Uganda at the time. So it's important showing how our knowledge of the mechanisms has been important for informing epi trials, and something similar is now happening in the Democratic Republic of Congo in another trial there. So essentially it's a 2-arm trial, but we have 2 different manufacturers providing nets. We have non-PBO nets from Olicet and their PBO Olicet Plus net, and from Vestergaard Fransen we have Permanet 2 and Permanet 3, Permanet 3 being the PBO treated net. So they were distributed randomly as part of the Universal Coverage Campaign. What you can see on the extreme right there is that there are approximately 16 million nets distributed to 104 health sub-districts across the Uganda, and those are also appearing in the sort of the coloured HSDs in the central map. We had balance between our PBO and non-PBO arms, around 52. HSDs received either PBO or non-PBO. And what we were doing is visiting the communities on a 6-monthly basis at baseline, 6, 12, 18, and 25 months. Looking at parasite prevalence in 10 enumeration areas, and so essentially that's 50 households, 5 households per enumeration area, and in 20% of those households we were also doing vector density measurements using Procopac sort of aspirators. Again, referring back to sort of Dave's talk, he's showing there that the parasite prevalence decreased across both arms of the trial. So we're looking at, as we move across this box whisker plot from baseline, 6, 12, and 18 months. And you can see it in the red is non-PBO and the blue is PBO. You can see that there's a marked fall in parasite prevalence at 6, 12, and 18 months. So there's still an impact of using nets. As Dave is saying, it may well be that they've lost their community effect, but certainly that personal protection, at least some degree of that personal protection remains. But what's key here is that you can see when you compare 6, 12, and 18 months, parasite prevalence is significantly lower in the PBO arms. And so just to put some numbers on that, if we look at parasite prevalences, so this is the ratio of prevalence in PBO versus non-PBO communities, And what we can see is that parasite prevalence fell by about 26% in those PBO communities, and that effect was seen at 8, 12, 18, and although the data aren't published yet, also at 25 months. So we do see a prolonged impact of PBO. We see much lower, or see significantly lower prevalences in those PBO users using communities. And just for those of you who have an entomological bent, this data was replicated by the Anopheles density. So again, same 6, 12, and 18 months time point, we saw between an 86 and a 75% reduction in the numbers of Anopheles resting in houses. So good evidence of entomological impact as well. So the results of this is essentially now we have the evidence from a second epidemiological trial of the enhanced benefits of PBO nets. So PBO overcoming P450s in an area of relatively high pyrethroid resistance, where we know it's mediated by P450s. There is a significant advantage to those communities who are using PBO nets versus non-PBO nets. And so now we have the second evidence base for an epi trial to move or help WHO move towards making that full recommendation around the use of PBOs. So with that, I thank you, and I pass on to Hilary.
Thank you, Martin. So next, please, Dave. So as a result of the work that we've been doing over the past 19 years, I think now, on building and synthesizing the evidence on the efficacy of PBO nets, or leading up to the development and the building the evidence base on the efficacy of the nets, and I work on mapping the distribution and the impact of insecticide resistance, and the combined with the work with WHO and other policy-making bodies. This has led to the deployment of PBO nets in Africa to combat pyrethroid resistance. So you can see here, this graph on the slide here shows the proportion of nets, the total net distributions deliveries to sub-Saharan Africa over the past 3 years. And in pink, that's indicating the proportion of those nets that are pyrethroid PBO nets. So approximately somewhere between 150 and 200 million nets are distributed to Africa every year. And in the first 3 quarters of 2020, 18% of those nets were PBO nets. So about 29 million PBO nets have been deployed in Africa so far this year to combat pyrethroid resistance. Now, pyrethroid PBO nets are seen by many as being sort of an interim solution, because as the intensity of pyrethroid resistance increases, there will likely come a point at which the PBO can no longer synergize the pyrethroids, and that's been shown in the models, and there's some evidence from laboratory studies, although not yet seen in the field. But they're seen as potentially a sort of stopgap while we get new nets on the market. And you can see on this graph here that in black, there's already some of those nets being used in Africa, and these are known as dual AI or dual active ingredient nets that contain pyrethroids. So all nets still contain pyrethroids because of their fast knockdown effect, but also a second insecticide. So it might be chlorphenapir in the case of Insecta G2 nets, or other nets have insect growth regulators on them. So there's already some new nets that are with more than one insecticide that are being used or evaluated in the field, deployed and evaluated as they're used. You can go to next, please. The work of LSTM, though, is looking to ensure the sustainability of the net market. So we have developed a pipeline for screening new compounds that are coming from industry partners and working closely with IVCC. So we have the Light facility that hosts the largest collection of pyrethroid resistant or populations of mosquitoes that are resistant to different chemistries, and so we can use these in vivo screens to determine whether the mosquitoes, whether these new chemistries will actually kill resistant mosquitoes. And then we have the in vitro screening pipeline that Gareth has already described, where we can look and see whether the individual enzymes that are elevated in resistant mosquitoes can metabolize these new chemistries and potentially cause cross-resistance. And then we have the transgenic mosquitoes, where we can actually test in vivo to see whether the new chemistries can confer cross-resistance to there's already existing cross-resistance. And this is also underpinned by an increasing number of behavioural assays that Philip's group have developed in order to see how the mosquitoes respond to these new chemistries, either alone or in combination with pyrethroid nets. So next, please. So this screening pipeline is proving to be very useful for industry partners, as it can provide this early warning system for indicating whether there's any potential cross-resistance to new chemistries. And it's actually already led to the changing in priorities of but from some of the leading insecticide manufacturers to change their lead chemistry and identify a more effective backup compound after following this pipeline and finding potential resistance mechanisms. So next, please. So I hope that these presentations today have sort of given a snapshot of the work in the research programs across the department, and shown how that we're working together to try and ensure that the future viability of bed nets, because bed nets, as you heard earlier from Dave, they've really proved to be one of the most effective non-pharmaceutical public health interventions globally. And it's really critical for the future success of malaria control that we can preserve the efficacy of this important tool. So the work from these multiple research groups across the department, only some of which have presented today, is really ensuring that we've got continued innovation in this space and using an interdisciplinary approach to working with stakeholders and with partners across the globe to maximise the research uptake and ensure that we've got continued new innovation in the bed net space. So finally then, the last slide, I'd just like to repeat Martin's comments at the beginning and to acknowledge members of the Vector Department, past and present. This work has been going on for many, many years, and it has involved a huge number of different staff in LSTM. And of course, also would not be possible at all without our large numbers of partners, our funders, but also key to this are many southern partners that we've worked with over the years to deliver on this research. So I'll end there and pass back to Martin.
Thank you. Thank you all the presenters, and we had a little bit of a technological glitch there. Hopefully you may have lost the presenter view. Hopefully that didn't impact your enjoyment. So we still have 5 minutes for questions. So if you want to ask a question, please edit it into the chat box, and we will do our best to get it to. Okay, so Graham Small has written in. Feel like a DJ. To what extent can transgenic lines expressing particular mechanisms of resistance be used not only to confirm the role of these mechanisms in cross-resistant, but also their fitness cost in the absence of insecticide selection? So I guess that's a question for you, Gareth, about the selective and fitness effects of these mutations in transgenics.
Yeah, thanks, Martin. Well, that's a great question, Graham. The transgenic lines, we're going to need quite a lot of optimization to look at fitness costs. So it's quite feasible these days to knock out individual genes. And so you could imagine knocking out some of the P450s and seeing what effect that has on resistance. But also you can do fine mutagenesis and change some of the target sites. And then just those small modifications, you could look at the fitness cost of those. At the moment, the lines we have overexpressed to a large extent, and so of the different P450s we've got. And so with those, they're going to have some fitness costs. which will be probably up and above what they have in the wild, where the expression is lower. And so you could fine tune overexpression to specific tissues, potentially to look at that. But that with the ones we have at the moment, it wouldn't really be feasible or fair to look at the effect of on fitness. But you could do something like that for target site mutations quite easily with CRISPR mutagenesis, which is what we're doing at the moment. So,
do you want to take on that and talk about observational studies in the wild and how you can look at adaptive around VGSE, for example?
So I think,
sorry, we got another question. So for Dr. Wheatman, do you know why the resistance in West Africa seems so much stronger and more prevalent? Is it down to increased bed net use, or is there something else?
So I don't think there's any evidence that it is actually down to increased bed net use or distribution. The bed nets are not the only selective force that has led to insecticide resistance or pyrethroid resistance. And certainly what preceded the onset of strong pyrethroid resistance, which was particularly notable in West Africa, although to some extent in Sudan as well, was really quite strong DDT resistance. So suggestion would probably be that there's been a relatively bigger impact of agricultural selection in West Africa with long use of DDT, given that it's very cheap and quite, for many years anyway, was quite effective in agriculture, selected for site mutation mechanisms in particular, which are very effective for DDT resistance, but also have carryover effects for pyrethroid resistance. Not as strong as for DDT, but gives that sort of leg up of resistance in West Africa, which has then been sort of taken on and expanded, particularly with metabolic resistance, by the distribution of pyrethroid nets. So I think it's almost more just that West Africa had a bit of a head start over others, rather than necessarily a greater selection from bed nets.
Okay, are there any other questions? We haven't got anything coming through on the chat. Okay, 5 seconds. Okay, so I guess it's almost 2 o'clock. So really just to thank you all for your attendance virtually, and thank all the presenters, and just to again fully acknowledge that this work is part of LSTM's collaborative efforts in developing translational tools for public health impact, and really just to thank all our partners, past, present, and hopefully future, both here in the UK and in the global south. So thank you all, and hopefully you enjoyed you found that informative. Thank you.
Thanks everyone.